So hello everyone. Now I think we are starting to have people checking into the webinar. And it's just 2 minutes past, so I hope that everyone will connect in the next couple of minutes. We have 96 participants registered in total, so we really much hope to see as many people as possible that will actually check in now. So today we will be talking about microbial biodiversity and focusing on soil samples. This is a joint webinar between Biomcare and the Danish Technological Institute. So I'll just slowly start with a bit of practical. So we are 3 people talking today. It's me, I will start, and then Helle and Valdemar will take over after about half an hour. So we're 2 people here from Biomcare and 2 people from the Danish Technological Institute. You probably have the ability to unmute yourself. I will ask you not to do so, and if you do, you will be muted again. So we ask you to ask your questions in the chat instead of speaking with too many people here to all talk. Yeah, I think that's all for the practical information. There will be one question I will ask during my presentation, but otherwise, I think due to the short time, we only have 1 hour in total. We will not do a lot of interaction beyond the questions. So today we have these 2 parts. We have the first part, which is my presentation about using DNA sequencing to study the soil microbial biodiversity. And we have the second part, which Helle and Valdemar will be talking about the soil biodiversity measurements in relation to sustainability and agricultural practices. We will organize in this way that you please ask your questions to me in the chat, and I will try to answer as many as possible within the first half an hour after my talk. And then for the questions you have to Helle and Valdemar, you ask in the chat also during their presentation, and then they will answer it afterwards. Okay, so for my part, this is focused on this method or technology that is DNA sequencing. That is a method that Biomcare is using. So Biomcare is a Danish company here in Aarhus that does everything microbiome, but we focus on this DNA sequencing and use it for biodiversity measurement. So I'd like to just share what that means. So I'll tell you about what it is, give you a short overview of the different types of DNA sequencing that can be used, why it's good for studying microbial biodiversity, and then how you can relate this to other informations about your samples that can make using DNA sequencing even more informative than other types of methods that you can use. So we talk about soil. It's not because soil is the only type of sample that you can have DNA sequenced and study biodiversity in, but soil is fascinating because it is one of the most complex microbial communities on Earth. It has thousands to millions of different microorganisms and covers different domains of life, which is a bit unique to other communities. And what is fascinating and challenging is that many of these microbes we have no information on. So they are unknown and uncharacterized and cannot be cultured. So there's a lot of information hiding in the soil microbiome. And at the same time, it is very important for agricultural practices, for crop production, for resistance to climate change and pathogenic diseases, and generally everything that we need the soil for. So it's really important and not well understood. Good reasons to work in this field. So with that sort of appetizer, we will dig into this concept of DNA sequencing. So sequencing is a method that we use to study the DNA, so the genetic material of microorganisms and any organisms really that has DNA. So in that way, it's different from the more traditional methods where you look in the microscope and you look at the shape of the cells, or you try to culture and by doing different staining and so forth, try to identify what type of microorganism you have. Using the sequencing platform, we take these complex microbiome communities directly and extract the DNA. And then we do some library preparations and put it on the sequencing machine, where we then identify what is the DNA sequences of the different microorganisms in the sample. So that's sort of the overall idea. I won't go into the technological about how they take the DNA from the sample and decode it into a file, so we have it on the computer, because there's different platforms out there, different technologies that uses different approaches. Kind of the end product or the idea is the same, that you take the DNA in the sample and you decode it into a file on the computer so you can work with it. Illumina is one of the most dominating on the market for short read sequencing, and then newer platforms for long read sequences is coming into effect. So part of the preparation, you actually chop the DNA into shorter sequences. So what you get is a lot of information about the code of short DNA sequences. And then I will be a little geeky and technical, but I think it's just gives you an idea about how this looks like, because what we then get from the sequencing platform is a file we call a FASTQ file that holds information about these DNA sequences. And that's the orange colored line here. It tells us, well, there was a DNA sequence in the sample that looked like this. Then there is some information about what sequencing machine, practical information, that's the yellow one, and then you have the quality. So how much do we actually trust that this first place in this DNA sequence was a G, when then the F tells us some level of confidence in that. And we use that with a lot of bioinformatics processing to decode and identify, well, which microorganisms was in the sample. And we relate it up to some large databases to annotate and say, well, do we know the name and any information about this microorganisms based on what other people have studied? And we calculate the abundance, so how much of each of these different microorganisms. And we can do that depending on the method, down to like from the phyla level or kingdom level and down to genera or species or even strain level, depending on method and complexity of the samples. So then we get this information. Here you see different phyla, it's color, different phyla microbes of color with different colors. And then we have a lot of species from each phyla in this round circle. It is a kind of a typical way to sort of show a taxonomic profile of a sample. And we use this taxonomic profile to calculate what we call biodiversity. And in this technical aspect, or when we analyze microbiome, we call it alpha diversity. So that's kind of one measure to show the complexity of the microbiome, of the microbes in the sample. There are different measures, different ways you can calculate, and they all represent different types of information, or see the diversity from different angles. So the most simple one is this observed, or called richness, where we calculate how many different microorganisms were there in the sample. And then you can make it a bit more complicated and say, well, were they present in the sample in the same amount? So we combine this richness with the evenness of the distribution of the microbes. And this is just sort of the two overall ideas. And there is a lot of different measures that you can calculate based on this, and then tell you a little bit of a different angle to it. I have included a slide here that tells you about some of the more common ones. And with this, I don't want you to read it now. I just want to let you know that there is a lot of this kind of too much information on my slides. So please email me afterwards or write me on LinkedIn if you would like to have the slide for you to sit quietly and read and get this information, or you actually want the recording of the webinar, then just let us know and we will send it to you. Because I don't expect you to read everything. This is a quick walkthrough of just the overall ideas, and then you are welcome to contact us later. So as I said, there's different sequencing methods. Beyond being different sequencing platforms and technologies, there's also different ways that you can work with the sequencing to get different types of information. And when you talk about microbiome, there's overall two methods. There is what we call the amplicon sequencing, where you in the library preparation target using primers a specific marker. So you, for profiling, for example, bacteria, you go for a specific gene, and then you get information detailed about the taxonomic profile of bacteria in your sample. The other common approach is called metagenomic sequencing or shotgun metagenomic sequencing. There you don't target a specific gene, but you try to sequence all the DNA that you can get in the samples. And the deeper you sequence, the more DNA you profile. And then you can actually get not just information about who is in this community, but also more functional information, which you can see here on the next slide. So you see, well, if you have the amplicon sequencing, then you select the marker, you get information on either the bacterial community, the fungal community, the archaea, or another group of microorganisms. And then you get the taxonomic profiles of these, and you often go down to like genera level. With shotgun sequencing, you can get more detailed information. You can have species or strain level information depending on the community you sequence, and you can have information about functionality. So you can have like genes and pathways. So this functional capacity of the microbiome profile. So it gives you a lot more information, but it also has some holdbacks, like it's more expensive than using amplicon sequencing. Often when you have, for example, a soil sample that is a highly complex sample, you have to use reference databases to study it. So you can only look at these microbes that we have information on to begin with. So it's not sensitive to unknown parts of the community. And that's probably how it's the most different, like from the amplicon sequencing, because if you use this, then your diversity will include the microbes that we have information on already. That's thousands of bacteria and hundreds of fungi, so it's still a lot of different microorganisms, but there's a lot we don't know about, and those we will not include in our calculation of diversity. If you use amplicon, well, you have to choose, are you interested in the diversity of bacteria? But then you can actually include in the calculation those we don't know the name of already, and include those to give you, so you will get a much higher diversity measure because you will include a lot of bacteria that we cannot annotate. So in that way, it has a different pros and cons in terms of what it can, and there's more information highlighted on this slide about why and when to choose either of these methods. So that gets me to a Q&A. So as I said, I'm running through a lot of information, and if you don't know about this to begin with, it can be a bit overwhelming. So we were thinking if there's an interest from you guys to have us host a workshop where we talk more in detail, maybe show you a bit how to calculate, how you work with this, what you can use it for. We would like to know that. So if you can let me know if you are interested, if you're not interested, or if you just don't really know. I give you a minute to answer that question. Okay, so I think we'll move on. So there's Okay, I'm getting a hold. I'm waiting.
Just another few seconds.
Okay.
Share results.
Yeah, so a good bunch of you, I think if I read this correctly, 63% of you guys would like to have a workshop on the subject, while 32% don't know and 5% are not interested. So I think that's a good bunch of people who think that could be interesting. I think we will put that into a schedule and come back with information on a workshop later this year, I think. So that's good to know. Thank you very much for sharing. And I think we will move on. I don't know. Oh, skal jeg trykke stop sharing?
Yes.
Jeg har den stadig kun. Jeg kan ikke få den væk fra min skærm. Jeg trykker det op, så...
Ja. Kan jeg se din skærm?
Kan I stadigvæk se resultaterne fra Poul? Nej, så er det bare mig. Det er fint. Det kan jeg godt abstrahere fra. Yeah. So why is sequencing a good method to study biodiversity? So I just talked a bit about the how we do it and what it means and the different methods. So one of the great advantages of using sequencing in general to study the microbiome, and especially for biodiversity, is the fact that you don't need to culture. So as I said, you take the sample, you take and extract all the DNA from the sample without needing first to do something, without having to culture. So the fact that we know so little actually about the soil microbiome means that using this method, we can also look at all the things we don't know, and we don't select for the culturable part of the community. So you will get a lot more diversity included in this when you use this method. It's largely unbiased, so you don't have to look in a microscope and say, well, I think this looks like. And it has a high resolution, so we often can go down to strain, to species level, and even to strain in some circumstances. So you can distinguish between microbes that often you cannot do with other methods. That can be at least very important if you look at the pathogenic aspects of microbes and so forth, that you can actually distinguish with this high resolution. Depending on the method, you can look across a branch of types of microbes, so the fungi and bacteria and parasites, so you can zoom in on one and look at only that. And it doesn't just give you this one measure of diversity, so you get a lot of information. You get this information of the relative abundance of hundreds and thousands of different microbes, and you can look at fungi bacteria ratios. You can look at other measures of diversity and relate it to other informations. You can look at the functional capacity if you choose shotgun sequencing. So in that way, you get this biodiversity, and then you get a lot more information that can help you to interpret. And that's what I will focus on for the remaining parts of this presentation. Because you can see here on this slide that you have one of the alpha diversity, one of the biodiversity measures called Shannon, listed on the y-axis of this graph to your left side. And then you have calculated these biodiversity measures for different soil samples coming from a corn field or a grass field, which has been really dry or really wet. And then the question is, well, does that affect the diversity of the microbiome? Well, you can see that the dry areas, independent of what crop there was, has a much lower biodiversity. So already there, now we're relating it to the metadata, to the study design, to like information about the samples. What do we know and what can explain the diversity that we are seeing? But we also would like more information, sort of. So what changed? What is actually this lower diversity in the sample? So we can zoom in with the information and say, well, let's have a look at the taxonomic profiles. Let's look if a few microbes spike, if they suddenly take up a bigger part of the microbiome in the sample and causing this lower diversity. And perhaps they have a certain function that is relevant for us. Or maybe it's relevant who we lost when we reduce the diversity. So we can actually sort of see, well, did we get a more simple community because a certain pathogenic organism suddenly took over this community? Or any other question related to that can help us to actually say, okay, so there's something to look at, then let's zoom in and learn more. And we can also do this data diversity, as we call it, sort of a community profile, and look like, is there a change in the overall community that explains how these samples relate to each other, how the diversity is affected in specific groups of samples? Are some of them more alike to each other than others? Do they look the same because they're coming from a certain field or treatment aspect? So then we can also add that information into your measure of biodiversity. And if you used shotgun sequencing, then you can also look at the functional capacity. I here just show one of our pictures of nitrogen metabolism, where we can look at different genes and how they are abundant or decreased in different samples. And we can relate this to the diversity we see, or to the study design that you have. So again, helping you to extract information about what is going on in the microbiome. Yeah, so this is just summarizing. So yeah, you do get this biodiversity, you can get this one number, and if that's what you need, and that's all you need to sort of work with your study, then that's what you can get. But you can then also get all these other informations and relate it to the metadata that you need or should collect from the samples that you're studying. And when you start about what you should and could do when you're doing this, well, there's some good practices about how to collect soil samples if you want to do sequencing, so that you treat it in the best way to then get the most true picture about how the actual community looked like in your sample. So we do have this free guide that you can go into Biomcare's website and download about how to collect samples and how to handle them depending on what you want to do with the soil sample. And then you can read about some of the limitations or suggestions about how to stabilize and store your samples and so on. And then just to round off my presentation, so I'm here talking on behalf of Biomcare, that is a company in Denmark that focuses on studying microorganisms, about using sequencing platforms to do all kinds of project support for people who are interested in the microbiome and doing its biodiversity or any other aspects of studying, if it can be cultures or samples, diverse communities in agriculture or livestock or anything of this sort. And this is just a short overview of our core services. So we also do a lot of consulting about how, if you want to get started with this, how you should do it. About the more complex communities, supporting a whole project, or profiling cultures, if you have cultures of microorganisms you want sequenced, or pathogens profiled or detected, and all these aspects about microbes and microbiome. So this is our contact information, and with that, I think I'd like to answer a few questions before I hand over the word.
Open up the chat.
The one above. First one, second one.
Okay, so I have a question from Kay Hestman, he writes, do you already have a standard criteria to test and see the quality of the microbial biodiversity of a soil sample? So to see if we think the biodiversity is high or low or good or bad or something like this. It is so dependent on the context of the sample, what kind of sample you have and what type of field it was, whether it's good or bad. We have a feeling, we're starting to have a good feeling about what we expect and what we don't expect. But for most situations, we ask people to send us some reference if they want to see if it's better or worse in something else, especially if it's from a part of the world or an area where we don't know so much to begin with, because for Denmark, we're starting to have a feeling. We have so from maybe Western Europe, but there's also many parts where we don't know what we expect. And then we would like to have this setup where you have, where you compare to something. So yes and no, it depends a bit. Then we have a question from Sharul Alam, who writes, what is FB ratio in last slide? So FB is fungi bacteria. So sort of to get an understanding about how the number of cells from these two types of microbes relate, if you have more or less fungi relative to bacteria in your sample. So this is based on DNA and cell count, because we count one cell in general in this approach. So it's a bit different from using biomass to study fungi bacteria ratio, but it's the same idea, just from a bit of a different angle.
1 minute.
There's from Johannes Greb, who writes, is it possible to perform shotgun analysis on multiple genes, or is it only possible to look at one gene at a time? With shotgun analysis, we do look at all the DNA at one time. We try to, if it's a more simple community, we can even assemble and regenerate the genomes so that we get a lot of information. With soil, that's not so possible, but we do look at all the genes, so independent so we're not interested in just zooming in on one gene. That's with the amplicon. There we look at one gene. You can do some mixing of primers to actually have more different regions in one sequencing run. But generally, we choose one at a time and then look at 16S for bacteria, ITS region for fungi, and so on.
Almost out of time.
Yeah.
It's another 5 questions.
There's one question here about the price range. It also depends on the method and the number of samples. So, but generally, if you want amplicon, it's about 100, little more than 100 euros per sample, and for shotgun it's about 200 euros per sample. So that's sort of the rough range that will be fine-tuned about with how much bioinformatics support and statistical support you want, and how many samples you have, and if you want DNA extraction also, or if you do that on your own. So I think that's all the question I will manage to answer, but please send me an email or write me on LinkedIn if you would like more, or if you have a project or something you want to discuss about using these methods, just reach out and we can have a discussion to follow up. And if you want the slides, let us know. So thank you for interesting questions and for your attendance. And then I'll hand over the word to Helle and Valdemar.
Thank you very much, Louise. And while we are just getting the technical part here to function, I can tell you all that we are very pleased to be here together with Louise from Biomcare. And the last two speakers of this webinar will be Valdemar, my colleague, and myself, and we are both from the Danish Technological Institute. The very shortest way of introducing DTI is that we work with foods, materials, and energy for a better future. I will start with just a few slides about the Institute. It is self-owned and it's a not-for-profit institution. We are a member of the Eurotech along with 9 other RTOs in Europe, and you may know some of them listed here. When we look at the UN's Sustainable Development Goals, our work at DTI centers around industry, innovation, and infrastructure, about responsible consumption and production, climate action, and zero hunger being the main issues here. And the Solgaard project that I'm going to talk about is very much related to both climate action and responsible food production. DTI in agriculture, just to finish off this presentation of our institute, is that we are specialists in field trials. Every year we design and perform around 1,000 field trials, and most of them are on farm. We measure greenhouse gases, and the last few years a setup has been developed for measuring nitrous oxide. We test tests and verifications of technologies, all different kinds, and in our research and development projects, we are very occupied with biomasses and use the optimal use of the side streams. Drones, digitalization, that's a difficult word for a Dane, and our AI, the other way around, is coming up really big now. And we also have specialists in software development and statistics. Enough about DTI, and now for Solgaard, this project that's under the European Union Horizon 2020 program. It's a 4-year project with 25 partners from 17 countries. And the key word here is soil biodiversity, and we'll look at it in relation to land degradation, soil management practices, and the soil ecosystem, its multiple functions and the services it provides for humanity. And finally, the effect of the climate changes on the soil biodiversity is also a big part of the Solgaard project. The aim of Solgaard, to put it in the shortest possible way, is to boost the sustainable use of soil biodiversity, to protect soil multifunctionality and increase economic, social, and environmental well-being. I will focus today on the field experiments that's the core of Solgaard. I will talk a little around soil biodiversity. And finally, I will put some emphasis on the dissemination. This also corresponds well with the soul of DTI, that we are a bridge between the knowledge institutions like the universities and the industry and the society. So it's about getting all the knowledge created out there in the world to do good. And here is the sort of concept overview of Solgaard. You can see the work packages here listed 1, 2, 3, 4, 5. And it all started with engagement, or that's still ongoing and will be throughout the project. Engaging farmers, consultants, stakeholders that are interested in conservation of biodiversity in the soil, politicians, and for a start, we have focused very much on the farmers as they were a prerequisite to start all the field work. That's the yellow part of this overview. We work here with land degradation gradients as defined by erosion, carbon content, and soil texture. And we have sampled soil in numerous sites across biomes and looking at different management practices. In all these soil samples, soil biodiversity has been detected, and we have also looked at many of the functions that the soil provide. All of this then is interlinked with work package 4, which is to create a valuation system to for all these ecosystem services. How do we value them? The food that we eat, of course, is obvious, but also all other aspects like the soil's capacity to store carbon or hold water, but also on the more socio-cultural part. How do we view the ecosystems? We all like to go out there in the beautiful forests and see the beautiful landscapes. How do we value all that? And all of that will then feed the work in work package 5, that is to develop a set of evidence chains. How is all this interlinked? So what cascade of effects will it have if you choose to shift your plowing of your fields to a reduced till or a no-till practice? What cascade effects will be initiated if you change from an annual crop to a perennial crop, or all these things that we can change? What effects will it have? And thus it all combines to some outcomes. The soil biodiversity and well-being framework it already is started, but it's a holistic approach to try to describe how all these different aspects are interlinked. Another outcome will be an app, a Soil Guardian app that decision makers can use to feed into the app what kind of practices you are currently using, and then the app will respond to you. What can you possibly do if you want to change the conditions for the soil biodiversity? We will address politicians and decision makers both nationally, in the EU, and even globally. So that's the short overview of the project. And coming down to earth now, what is actually going on? In the work package 2, we have been sampling soil from 234 sites in 10 countries. The overview here, I think we'll go to the work package 3. So that's the cross-biome network of sites is one part. And in the work package 3, we are simulating climate changes. The cross-biome refers to the fact that we are working with croplands and with grasslands in Ireland and with a forest in Finland. And on the map, you can see the European countries involved in this. Last year, we simulated drought in both organic and conventional fields. And this year, we will repeat that, creating a 3-month drought period, but also combine it with a 5-day heat wave that aims at rising the temperature with about 5 degrees. And of course, we have controls for all of that. So these climate simulations are taking place in 7 countries, and we will be sampling soil at different times. And altogether, we end up with probably around 600 soil samples. All these soil samples are sent to universities and institutes in Netherlands and Spain primarily, and they will all be analyzed for a range of things that I'll come back to in a little while, because I forgot to introduce you to the Danish contribution. Last year, we were sampling soil in 30 spring barley fields, comparing conventional fields to young organic fields to older organic fields that have been grown organically in more than 5 years. All these fields were situated in the eastern part of Midtjylland, as well as the island of Funen. And we have the yellow here mark that's sites of medium land degradation, and the red ones are high land degradation. So we will be comparing that. And the climate change simulation, you can see here, we are putting up these shelters in the fields, and they are left there for 3 months. And inside half of the shelters, we will, in July this year, put up a ring of heaters to create the heat wave. And now to the analysis performed on these many hundred of soil samples. The bulk density, a range of physical and chemical properties, the DNA analysis that I'll come back to, and very much like what Louise has described. For the climate simulation, we will also look at the rhizosphere. Microscopy of nematodes, mites, and springtails will be compared to the DNA analysis of these groups of organisms. And finally, we will be looking at litter decomposition using the T-bag index. I really like that because it's so simple. And for those of you who don't know it, it's recognized internationally as a way of measuring decomposition or comparing different soils. We simply bury into the soil tea bags of green tea and rooibos tea, leave them in the soil for some weeks, and then we take them back to the laboratory, weigh them, and the weight difference then is a measure of the soil's ability to decompose the litter. There are lots of other indicators of soil functions that I haven't listed here, but to give you an overview of what we are doing. Yeah, sorry. Okay, then for the DNA, to assess bacteria, archaea, protists, nematodes, collembola, mites, and earthworms, we are assessing 3 ribosomal markers and 1 mitochondrial marker. For the vira, we are using the shotgun method. And to assess the abundance of these different organisms, as well as the mycorrhiza communities, we are using quantitative PCR, as well as neutral and phospholipid-derived lipid fatty acids. And finally, we will look at the nitrogen cycle, again using the quantitative PCR to assess the abundance of these functional genes. At each of the sites in the climate simulation experiments, these undisturbed soil cores were taken out and shipped to Amsterdam for microscopic assessment of the nematodes, mites, and springtails. And the last here is that for a subset of samples, 15 to 100, we have tried to sequence DNA from 10-gram samples and compare that to the 0.25-gram samples that is ordinarily used. Okay, so this was trying to go quite quickly through what we are working with in SoilGuard. to sum up, the field experiments are to provide us with a better understanding of soil biodiversity as a function of land degradation, of soil management practice, and of climatic conditions. Also, we aim at making contributions to the standardization of the methodology in taking soil samples for these kind of analysis. The soil biodiversity, well, the complexity is great, and we do need to try and get an idea of how everything is interlinked. So the SoilGuard project addresses the links between soil biodiversity and soil management practices, between the soil multifunctionality and the human well-being. The modeling work will also, of course, aim at increasing the power to forecast soil biodiversity responses. Finally, the dissemination will be going on in writing as well as in events like this, being the first one from our side. We will create this SoilGuard's network of knowledge with all people interested, and an app will be created that you can feed this app with knowledge on the appropriate piece of land, how it is managed, and the app will give response. We will address politicians, and we will address conservation stakeholders and farmers, and lots of activities will be going on starting from now. And with the results from all the analysis beginning to come in this year, there will be lots of opportunities to actually learn about the results. So if you would like to be part of the SoilGuard network, please write your email in the chat now. This way we can get back to you, and you will be given information on what's going on in SoilGuard for the next coming years. And while you do that, those of you who would like to join us, I just want to take the opportunity to say thank you to my colleagues from DTI that have been involved in SoilGuard, and also a big thank you to all our partners in the SoilGuard, especially the group of people that we have been working with in the climate simulation work in the field. It has been and is a privilege. So a big thank you from here. And then I think it's time to make a shift again, and I will hand over the floor, so to say. I will hand over the screen to Valdemar, who will take you up in the helicopter and talk more about the bigger perspectives of assessing soil biodiversity. Thank you very much.
Hi, I'll just find my own slides. They'll respond. So I'm here to ask the big questions. Why are we doing this? Why does it matter that we can measure biodiversity in soil, and what are we supposed to use that information for? So why it's important is basically, or part of it is, the ecosystem services that soil provides us in agriculture. So ecosystem services is things that our natural environment does for us, or the environment in general. That is like areas of wetlands that deal with flooding, or it can be just the cultural things that we can enjoy by nature. And the soil under our fields also serves a lot of services for us.
Sorry, Valdemar, are we supposed to see your slides now? Because
Yes.
we are still seeing the previous. So I think you just share, you didn't share. Yeah, then we are able to see. Perfect, thanks.
All right, it's done now. Is it on? The big Y or but Y?
Yes,
Good.
it is.
Thank you a lot. So you didn't miss much. This is the important first slide. So here are some examples of ecosystem services that soil and agriculture do for us. One of them is carbon storage. Carbon sequestration is a big topic in agriculture now, how we can use the soil to store carbon. Fungi aid in uptake of nutrients for plants. It's also the microbiome in the soil and how it deals with nitrogen and other nutrients is important for nutrient leaching, and how nutrients that's supposed to stay on the field ends up in the environment around the fields, like in wet waterways and situations like that. Then there's the plant health. There are pathogens in the environment, and how the other microorganisms present in the environment has an effect on how those pathogens affect plants. And general soil health, like we know from earthworms digging in the soil, aid in aeration of the soil, which is also good for the plants. So these are few examples of services that the soil do for us. And therefore we are, and not just the soil, the organisms in the soil. That's why we are interested in know what they're doing, why, when, what effects we have on them, how we can change them, and so on. So I like to dig a little bit deeper into the carbon sequestration part of it. So in Denmark, we have this big experiment or survey that's called Kvadratnet, which is sampling agricultural soil for all over Denmark. There's thousands upon thousands of samples, and one of the things they're looking at is carbon. So the most recent results haven't been published yet, but I was at a conference a week ago where they shared some of the results. And they say we have about 100 tons of carbon per hectare in agriculture in Denmark. With how much agricultural land covers in Denmark, and the capacity for carbon storage in them, if we can just increase 1 percentage point of how much carbon is stored in soil in average in Denmark, in our agricultural, that's the same as 20% of the carbon reduction we're supposed to do every year from 2030 and onwards in relation to the governmental goal of lowering our total agricultural emissions by 60%. So it's just to say that there's a huge pool, and it's not like this is the solution to climate change in Denmark. That's not what I'm trying to say, because the microorganism and how they take up carbon and store it, it's a balance. You can't just add carbon to the soil forever. At some point you reach an equilibrium. It's just to say that it's a huge capacity, and if we could increase it by maybe just 2 to 5% overall in agriculture, we would remove a lot of the carbon that's already in the atmosphere. And that would be great, as long as we can also lower it, how much we emit. Another reason why it's important to look at soil biodiversity is that it's really complex. So what I have here on the right of the screen is a food web of organisms in the soil. And of course, there's bacteria here, and they're represented by a whole bunch of different bacteria. There's nematodes, there's earthworms, so on, and different kinds of mites, and all sorts of fungi, and all these organisms that each do something in the soil and is part of this system that works together. And that's why how complex this is a reason why it's interesting to look at it with genomics, and we can look at a lot of them, because if we try to isolate each one of them, it's very difficult to see what's going on. We can either try to optimize for biodiversity in general and say we want a diverse microbiome or organism society in the soil, and assume that as long as it's diverse, each of these niches are represented, or we can try to optimize for each one of them, which, I mean, the figure should show why that's perhaps a poor choice. Another reason why we should care about biodiversity is that it's perhaps not that well understood. So one of the services that the organisms in the soil do for us is nutrient management. They take nitrogen that we add into the soil and make it available to the plant, and then take excess nitrogen and release it as free nitrogen again. The nitrogen cycle, as everyone has probably, as a lot of us has probably been taught. But the thing is, the nitrogen cycle here with denitrification and nitrogen, ammonia oxidation and nitrogen oxidation and so on, it's not the full picture. Just in the last 30 years, we realized that the figure looks like this instead. We learn about aminox, and we're starting to learn how important it is. Commanox is a very recent addition to this graph. I think it's like 2013 that was discovered or something like that. It's just to say that perhaps there's processes in the soil that we still don't understand or know about. So when we're trying to look at the biodiversity in the soil, it isn't necessarily always necessary to look at it functionally. We can just look at the environment that is there and the ecosystem that is there and try to protect the diversity as it is, so we don't end up losing something that we didn't realize was important. And why do we care about losing things? So the situation is that we might be dealing with the consequences of climate change. And the fear in that is when we're going from the slush field on the left to the more drier field on the right, is that we still want to be able to farm that land. And to farm that land, we are reliant on the services that the soil organisms provide for us. So we don't want them to disappear. And that's a real fear. But the fear becomes bigger the less diverse the soil is. If we have 3 earthworms in the soil, there's a good chance that one of those earthworms or 2 of those earthworms can deal with a changing climate and it becoming drier or hotter or whatnot. While if we only have 1 earthworm, we're really vulnerable to change. If that one species we have in our field can't deal with the increased heat, we don't have that services provide us anymore. So that's why we care. That's why Soil Guard is put in place, to look at the biodiversity, how it's influenced by the climate change and different agricultural practices. Yes. So that was for me. And we will provide you with the slides if you want them. You can just message me. I have all these sources for what I was trying to sum up. It's a difficult challenge to has 7 minutes to sum up what some would call an entire field of science. It's a big subject I'm trying to deal with. So of course I can't do it much justice. So
I think time is almost running
yes.
up, so maybe we have time for 1 or 2 questions. Can we see the questions here, or do we need Regin to read them out to us?
You can read out the questions loud. I'll send it to you in a direct message, so I'm sure that you have them.
Yes, we are asked if there's a living lab in Soil Guard.
Not as that, no. We hope there will be, but not at the moment.
And we are asked if it's volunteering to be in Soil Guard, and I guess it is.
Yes, it is.
Yes.
Yeah, everyone can volunteer to be part of the Soil Guard network of knowledge. And all it means is that you will be given information when there are events, or maybe you will be asked to answer surveys. Yeah, it's underway, and I don't know at the moment what's going to happen during the next 2 years. You can always join up, and if you don't think it's interesting, you can pull out again.
Yes. And Boris asked me, I guess, about the limits of carbon sequestration. What's the fundamental thinking behind the assumption of the limits? So there is a limit. Of course, you can't store more than 100% of the soil can't be carbon. So there is the fundamental limit that there is a volume, and that's what there is. And there's also other things in the soil past carbon. But the thinking is there are a lot more studies of this in England where they're studied for a longer time. The problem with carbon sequestration studies is that it takes very long time to store anything. So it takes a long time for you to see changes based on your experiments. But what they have shown is you get to a point where you can increase it, and then you stop seeing the increase. And we also know about the potential for it. We can see at other environments, forests are better at storing carbon than agricultural land is, and they store upwards of 80% of the soil is straight up carbon, or carbon based molecules. So that's probably the limit, theoretical limit. I guess the limit in agricultural, it really depends on the soil type, if it's sandy or silt or what it is, but it's maybe in Denmark theoretically possible to get an average of 60% maybe. But that would also probably include some biochar.
I think that the time is up and there are more questions, but we have them, and you are very welcome to contact us afterwards, or we can get back to you. We will keep all your comments and your emails, and we will join with you later.
Yes.
We should give the word back to Louise if you want to round off. We can say thank you from here.
Yes.
Yeah.
Yes, and I'm back, and thank you, Valdemar and Helle. That was very interesting. And I think there's only just left to say thank you very much, everyone, for listening in. Please be in contact if you have any questions or comments or interests. You can always write both me and Helle and Valdemar and Regin and anyone in these organizations here, that we will get back to you. If you want the recording or the PowerPoint, let us know. And then I think we will work on a workshop on the subject to get more into detail. There was also some comments about limitations in terms of how you get relative abundances versus real absolute abundance, and if you can combine the methods with some qPCR or spike-in methods that we also do, and we can spend a lot of more time on. And there was like comparing the nanopore with Illumina and how that looks like for the future. So if we want a lot more detail and technological discussions about what is possible and what is not, and what we know and what we are working on, then keep an eye on our updates, and we will put together some more information either in webinars or workshops in the future.